Hello students, I hope that all of you are doing good. So I hope that your preparation it is going fine. And so in this video lecture, we would be going through the revision of the following 4 chapters that are present in the Unit 1 for the Biology Entrance CUET test. That is chapter 1, that is reproduction in organisms. Then we would be revising the important concepts of sexual reproduction in flowering plants. After that, we would be learning about the human reproduction important concepts. Then we would be going through the reproductive health. So before starting this video lecture, we would just be discussing some tips that you need to follow right now before the few days of the entrance test. So first of all, now we need to revise all the important concepts that are present in the chapters. So first thing that we need to do is that we need to do rigorous revision of all the key concepts. So these key concepts that would be discussed in this video lecture. So after the revision, we need to try to recall whatever we are learning. So whatever we are learning, we need to recall. And after that, we need to practice as many questions as we can. So we need to practice many multiple choice questions so that we are prepared in a very good manner. And also we must not now study any new concepts. So whatever we have learned, that is enough, and we need to revise it again and again. So now let's start with this video lecture. So first of all, in the chapter of reproduction in organisms, we need to know the difference between the asexual reproduction and the sexual reproduction. So the first most major difference between the asexual and the sexual reproduction, it is that in asexual reproduction, only a single parent is involved. Whereas in the case of the sexual reproduction, 2 parents are involved. So the major difference between the asexual and the sexual mode of reproduction, it is that the formation of gametes doesn't happen. The formation of gametes, it doesn't happen in the case of the asexual reproduction. Whereas formation of the male and female gamete, it takes place in the sexual reproduction. So as there is no formation of gametes in the asexual reproduction, there is no fusion. No fusion of gametes takes place in asexual reproduction. Whereas in the case of sexual reproduction, there is fusion of the male and female gametes. So the asexual mode of reproduction, it is happening in the case of amoeba. That is, they reproduce asexually by the mode of the binary fission. And also budding, it happens in the yeast. Whereas the sexual mode of reproduction, it is being done in the case of human beings. Right. So this slide, it is very important as it is telling us the difference between the asexual and the sexual mode of reproduction. So now let's move on to the next key concept from this chapter. So now we need to learn about the types of asexual reproduction. So a multiple choice question can be asked from this. So we have the following 4 types of asexual reproduction, that is fission, budding, sporulation and fragmentation. So in fission, what is happening is that the cell, it divides into 2 halves, and each half it is getting, then it becomes a adult. So this happens in the case of amoeba and paramecium. Whereas budding, it is happening in the yeast, where a bud, it starts to arise from the parent yeast body, and then it is getting separated and it forms a completely new organism. So budding, it happens in the case of yeast. So in the case of sporulation, the spores are formed. So the spores, they have very thick walls that helps them to survive in the extreme environmental conditions also. So these spores, they are present in the plants and fungi. So fragmentation, it is done in the hydra, where the parent body of the hydra, it is dividing into fragments, and each fragment it is getting raised into a new, completely new organism. So now let's move on to the next concept. So vegetative propagation. So this, in this type of asexual mode of reproduction, a completely new plant, it is getting arise from the vegetative parts of the plant. So what are the different vegetative parts of the plant? Let's go through this diagram. So here we can see that some eyes, they are present in the case of potato. So if I cut this slice of potato which has the eye, and I put it in the soil and I give it water and nutrients, so definitely the new potato plant, it would arise from this. So the vegetative part that is present in potato, it is the eyes. In the same manner, the vegetative part that is present in ginger, it is rhizome. Whereas the agave, it consists of the bulbils, the bryophyllum, it consists of the adventitious buds, and the water hyacinth, it consists of the offset. So all this, the eyes, the rhizome, the bulbil, the bud, and the offset, all these are the vegetative part of these plants that helps in the giving rise to new plants. So now let's move on to the next question. So sexual reproduction. So the sexual reproduction, it is getting divided into three parts. That is pre-fertilization. So the events before the fertilization process, it happens, it is known as the pre-fertilization. Then we have the fertilization process, and after that we have the post-fertilization process. So now let's go through the pre-fertilization that happens in the case of the sexual reproduction. So gametogenesis, it is defined as the process of the formation of gametes. So gametes, they are haploid in nature. So this is very important, that gametes, they are haploid in nature. That means that they only consist of one set of chromosomes. So now let us understand the difference between the isogametes and the heterogametes. So isogametes, they are gametes that are similar morphologically and physiologically. Whereas heterogametes or the anisogametes, they are different in morphology and physiology. So the examples of organisms that produce the isogametes, they are Cladophora and Eulothrix, whereas heterogametes or the anisogametes, they are produced by the algae, fucus, and the human beings. So in the human beings, we know that there is this ovum that is stationary, and we have the sperm that is motile. So both of them, they are morphologically and physiologically, they are different. Also the ovum, it is large in size because it consists of the cytoplasm amount in the ovum, it is more. That is why it is large in size. Whereas sperm, it is small in size, as the cytoplasm amount in the sperm, it is less. So that is why they are an example of the heterogametes or the anisogametes, as they are morphologically and physiologically different. So now let's move on to the next concept from this chapter. So now the important concept in the pre-fertilization, it is what is meant by bisexual organisms and what is meant by unisexual organisms. So bisexual plants, they have both the male and female reproductive structures in the same plant. So the keyword, it is both male and female reproductive structure, they are present in the same plant. Whereas in the case of the unisexual plants, both the male and female reproductive structures, they are on different plants. So the keyword here, it is they are on different plants, right? So the bisexual plants, they are also known as homothallic and monoecious. Whereas in the case of unisexual organisms, they are also known as heterothallic and dioecious. So the examples of the bisexual plants are hibiscus, rose, and sweet pea. Whereas in the case of unisexual, we have papaya, watermelon, and corn. So now let's move on to the next key concept from this chapter. So now, after the pre-fertilization process, the next process that is happening in the case of sexual reproduction, it is fertilization. So in the case of fertilization, what is happening is that the male gamete and the female gamete, they fuse together and they form zygote. So fertilization, the types of fertilization, it is two types, that is external and internal. So in external fertilization, the algae, fish and amphibians, the syngamy, it is occurring outside the body of the organisms. That is, the fusion, it occurs outside the body of organisms in the case of external fertilization. Whereas in the case of the internal fertilization, it is occurring inside the body of the organisms in plants such as fungi, mosses and pteridophytes, whereas it also happens inside the fusion, it is happening inside the body in the case of reptiles, birds and also mammals. So the male and female gamete, they fuse in the case of fertilization. So after fusion, what is formed is known as zygote. So now let's move on to the next concept from this chapter. So now the next process that is happening in the case of sexual reproduction, it is the post-fertilization events. So this happens after the fertilization process, it has happened. So after the fusion of the male and female gamete, that is also known as fertilization, the zygote, it is formed post fertilization. So this statement, it is a very important statement. So let's go through it. It is the vital link, zygote, it is the vital link that ensures the continuity of species between organisms from one generation to the next. So embryogenesis is also a post-fertilization process in which the development of the embryo takes place from the zygote. So now let's differentiate between the two types of animals, that is the viviparous and the oviparous. So what happens in viviparous animals is that the development of the zygote, it is taking place in the body of the female parent. Whereas in the case of the oviparous, so oviparous animals, they are also known as the egg-laying animals. So hard shells of the eggs render the protection, and once the fetus, it is maturing, the egg, it gets hatched. So in the case of viviparous animals, the chances of survival, it is more. Whereas in the case of oviparous, the chances of survival are less, because the development of the zygote, it is taking place outside the body of the female, right? So the survival, the chances of survival, it is less, because the attacks from predators, it can happen, and thus the survival chances, they decrease. So vivipary, it is taking place in human beings and also in dogs, whereas ovipary, it is taking place in the reptiles and birds. So now let's move to the next concept. So in the post-fertilization process in the plants, the following plant parts, they get developed into this following parts of the plant. So now let's go through the development processes. So in the plant, the zygote, it gets developed into the embryo. So the ovule, it is getting developed into the seed. The integument of the ovule, that is the wall of the ovule, it is getting made into the seed coat. So ovary, it is getting matured into fruit, and ovary wall, it is making the pericarp, right? So now let's move on to the next slide. So we have discussed all the important concepts from the chapter 1. Now let's go through the chapter 2, that is the sexual reproduction in flowering plants. So the flowering plants, they are also known as angiosperms. So sexual reproduction, it is the process in which new organisms, they are made by the fusion of the male and the female gametes. So the flower, it is the primary reproductive structure. This is very important. The flower, it is the primary reproductive structure, and within the flower, the reproductive organs, they are being produced. So now let's go through the male and female reproductive organs. So the male reproductive organ, it is also known as the andrecium. So andrecium, what is it made up of? So andrecium, it is made up of stamens that consist of the filaments and the anther, and this anther, it consists of the pollen grains. So the female reproductive organ, it is also known as the gynecium. So this gynecium, it is also known as pistil, and this pistil, it consists of the stigma, style and the ovary. So now let's move to the next key concepts from this chapter. So now let's go through the pollen grain, that is the male gametophyte of the plant. So a question can come, which is the male gametophyte of the plant? We know that it is the pollen grain. So the pollen grain, it is made up of two layers. So the outer layer, that is hard, it is known as the exine, whereas the inner layer, that is soft, it is known as the intine. So why the outer layer, that is the exine, it is hard? Because it is made up of the sporopollenin. So this sporopollenin, it is one of the most resistant organic materials. So it can withstand the high temperatures and also the strong acids and alkali. Because it can withstand all these environmental conditions, the pollen, it can be preserved for long years. So there are some apertures that are present in the exine, and these apertures, they are known as the germ pores, where the sporopollenin, it is absent. So this is a very important point, that where the sporopollenin, it is absent. So it is absent in the germ pores. So now let's go through the inner layer, that is the intine. So the intine, it is made up of cellulose and pectin. So cytoplasm of the pollen grain is surrounded by the plasma membrane. So here we can see the structure of the pollen grain, and here we have the hard outer covering, that is the exine. Now here we can see that some apertures are also present, and these apertures, they are known as the germ pores, where the sporopollenin, it is absent. So now let's move on to the next concept. So this slide, it is a very important slide, and this is a very important concept of the female gametophyte, that is the embryo sac. So the female gametophyte of the plant, it is the embryo sac. So this diagram is also very important, and we need to learn this diagram. So now let's go through the points from this slide. So let's go through now the diagram. Let's understand it through the diagram itself. So here that we have two ends of this embryo sac, that is the chalazal end and we have the micropylar end. So you can get confused that which end is which. So a better way to learn this is that the A, C. So this A, it is indicating the antipodals, and C, it is indicating the chalazal end. So now we know that the micropolar end, we have the synergids and the filiform apparatus automatically. So in order to, you do not get confused, you can remember it like this, that AC, the A here stands for the antipodal and C is standing for the chalazal end. So the chalazal end, it consists of the antipodals, the micropolar end, the micropylar end, it is consisting of the synergids and the egg cell. Okay. So now this embryo sac, this mature embryo sac, it is known as the 8 nucleate and 7 cell. Why it is known like that? Let us go through it now. So we have 1, 2, 3, 4, 5, 6, 7, 8. So we have total of 8 nucleus in this embryo sac, and here now why it is known as 7 cell, let us go through it. So 1, 2, 3, we have 3 cells here. Now this 2 nuclei that is present, that is known as the polar nuclei, it is considered as a single cell. So this single cell, it is known as the central cell. So this single cell, it consists of the 2 nuclei. Then we now total we have 4 till here, 4 cells we have. This is the 5th, 6th and 7th cell. So that is why it is known as 8 nucleate and 7 celled, because this central cell, it consists of the 2 nuclei. Okay. So now what is happening is that this filiform apparatus, it helps in the entry of the pollen tube inside the synergids. So now let's move to the next key concept. So double fertilization. So sure shot from this topic, a question will come in the exam. So now let us understand the double fertilization process that is happening in the case of the angiosperms. So what is happening in the case of double fertilization is that here we have the pollen grain. So this pollen grain, it is received by the stigma. So this is the female reproductive part of the plant, that is the stigma, style and ovary. So here we can see that the stigma, it is sticky in nature and thus it receives the pollen grain. So now this pollen grain, it consists of the two cells, that is the generative cell and the vegetative cell. So what is happening is that the generative cell, now what is happening in this step is that the generative cell, it is getting divided into the two sperms here, and we can see that the pollen tube, it is elongated. So what is happening is that this pollen tube, it is then elongated, and what happens is that this pollen tube, when it is getting elongated, it fuses inside this ovule. So it is getting fused inside the ovule by the micropyle, that is the opening that is present in the ovule. So now the pollen tube, as it penetrates the ovule through the micropyle, what is happening inside this ovule? So this is the enlarged structure of the ovule. So here we can see that this is the micropylar end where the egg and the synergid is present, and here this is at the chelazal end we have the antipodals. So the two sperm cells, so from that one sperm cell, it fuses with the egg here, and the other sperm cell, it gets fused with the polar nuclei that is present in the central cell, right? So in this case, we see that the two sperm, they are getting fused two times. So that is why it is known as the double fertilization process, because fusion of sperms, it is taking place two times. And after that, what is happening is that when the sperm cell, it is getting fused here with the polar nuclei of the central cell, it is known as triple fusion. It is known as triple fusion because we can see that three nuclei, they are getting fused with each other. So one nuclei, it is of the sperm, the two nuclei, it is of the central cell. So that is why this fusion, it is known as the triple fusion, and the process, it is known as the double fertilization process. So this concept is very important, and we must know each and every detail of this concept. So now let's move on to the next concept from this chapter. So now let's go through some important points. So first of all, let us understand what is meant by a true fruit. So the fruit that is getting developed from the ovary itself, it is known as the true fruit. But when the development of the fruit, it happens with the thalamus, it is also taking part in the development of the fruit along with the ovary, then it is known as the false fruit. So thus the examples in which the false fruit it is being produced is in the case of apple, strawberry and cashew, in which the thalamus also takes part in the formation of the fruit. So fruits such as banana, they develop without the process of fertilization. So that type of fruits, they are also known as the parthenocarpic fruits, because the development of fruits, it happens without the fertilization, and these fruits such as banana, they are generally seedless. So now let's move to the next key concept. So apomixis and polyembryony. So these concepts are also very important from this chapter. Let's go through it. So the species of Asteraceae and grasses, they have involved a special mechanism where the seeds, they are produced without the process of fertilization, and it is known as apomixis. So don't get confused between parthenocarpy and apomixis. Parthenocarpy, the fruit, it is developed without the process of fertilization, whereas in apomixis, the seeds, they are produced without fertilization. So the occurrence of more than one embryo in a seed, it is known as polyembryony. So the name itself, it is suggesting that poly, it means that more than one. So more than one embryo, if it occurs, it is known as polyembryony. So now let's move on to the next concept. So we have discussed all the key concepts from the chapter 2. Now let's go through the concepts of the chapter 3, that is human reproduction. So in human reproduction, the male and female reproductive parts, you can go through it. So now let us discuss the very important concept from this chapter, that is gametogenesis. So gametogenesis, it means the formation of gametes in the gonads. So gonads, it means the primary reproductive sex organs in males and females. So the types of gametogenesis, it is the spermatogenesis, that is the formation of the sperms, and the oogenesis, that leads to the formation of the ova. So now let's move on to the next concept. So spermatogenesis, what does it mean? So it is the process of the formation of the sperms in the seminiferous tubules of testes. So this is very important point, that the formation of the sperms, it is taking place in the seminiferous tubules in the testes. So there are two stages of the spermatogenesis, that is the formation of spermatids, that is the first stage, and the second stage, it is the spermiogenesis. So what happens in the formation of spermatids, it is that the sperm mother cells, that is the spermatogonia, they get formed into the spermatid. So in the second stage, what happens in the, that is known as the spermiogenesis, which is the second stage of the spermatogenesis, is that the spermatids, they transform into sperm. So now let's go through the next concept. So what is happening in spermatogenesis? Let's go through the following steps. So it is very important to go through these steps, as arranged in chronological type of questions can be asked from this. So first of all, the spermatogonia, it is diploid in nature. So 2n, it is indicating diploid, and n, it is indicating haploid. So it undergoes mitosis, and after mitosis, the primary spermatocytes, they are formed. After the formation of the primary spermatocytes, the meiosis 1 is happening, and it leads to the formation of the secondary spermatocytes. After that, the meiosis 2, it happens, and it leads to the formation of the spermatids. Then the spermatids, after the process of spermiogenesis, it leads to the formation of the spermatozoa, that are also known as the sperms, and they are haploid in nature. So after the spermiogenesis, like after this step, the formation of the spermatids, the sperm heads, they become embedded in the Sertoli cells, and they are finally released from the seminiferous tubules by the process which is known as the spermiation. So don't get confused between this spermiogenesis. So in spermiogenesis, what is happening is that the spermatids, they are getting converted into the spermatozoa, that are also known as the sperms, and they are haploid in nature. Whereas in spermiation, the sperm cells, they get embedded into the Sertoli cells, and the sperms, they are finally released from the seminiferous tubules. And that process of the release of the sperms from the seminiferous tubules, it is known as the spermiation. So now let's move to the next concept. So it is very important to know the role of hormones in the spermatogenesis. Also, in the case of the role of the female hormones, that also we need to study. That we would be going through that in the coming slides. So first of all, let's go through the role of hormones in the formation of the sperms. So the hypothalamus, it secretes the gonadotropin releasing hormone, which acts on the pituitary gland. So this pituitary gland then secretes the two gonadotropins, that is the luteinizing hormone and the follicle stimulating hormone. So the role of both these hormones, it is very important in this chapter, and a short question from the role of hormones, it can come in the exam. So the luteinizing hormone, it is acting upon the Leydig cells. So luteinizing hormone, it acts upon the Leydig cells, whereas the follicle stimulating hormone, it is acting upon the Sertoli cells. So when it acts, the luteinizing hormone, it acts upon the Leydig cells, it leads to the secretion of the male sex hormone that is known as the androgen. And this leads to the stimulation of the spermatogenesis. So we have learned that the spermatogenesis, it is of two parts, in which in the first part, the spermatogonia, it becomes the spermatid, and then after that the spermatid, it becomes the sperm. So the luteinizing hormone, it is acting upon the Leydig cells. The Leydig cells, they are responsible for the secretion of the male sex hormone, that is androgen, and this androgen, it leads to the process of stimulating the process of the spermatogenesis, that is the formation of the sperms. Whereas the follicle stimulating hormone, it is acting upon the Sertoli cells, and the Sertoli cells, then it stimulates the secretion of the factors for the spermiogenesis. So what is spermiogenesis? So the conversion of the spermatid to the spermatozoa, that is the sperm, it is known as the spermiogenesis. So that is it is then the secretions that are secreted by the Sertoli cells, it leads to the process of this spermiogenesis. So now let's move on to the next concept. So now let us study the structure of the sperm. So the main parts of the sperm, it is the head. So this head, it consists of a cap-like structure. So this cap-like structure, it is known as the acrosome. So this, it is very important, the role of acrosome. So the acrosome, it is filled with the enzymes. So this enzymes, it helps in the fertilization with the ovum. So the role of acrosome is that it consists of enzymes that helps in the fertilization of the ovum. So the middle part here, it is consists of the mitochondria. So the mitochondria, it is acting as an energy source of the sperm that helps it in swimming. So the tail, so this portion, it helps in the motility of the sperm. So now let's move on to the next concept. So now let us understand the formation of the ovum, that is the oogenesis, the formation of the female gamete. So what is happening is that the oogonia that is diploid in nature, it undergoes mitosis and it leads to the formation of the primary oocyte. So the primary oocyte, it undergoes unequal division. So this unequal division, it leads to the formation of a large size secondary oocyte and the first polar body. So the role of the first polar body, that what happens with it, it is quite unknown, that whether it divides further or it gets degenerated. So after that, the formation of the secondary oocyte and the first polar body, the Graafian follicle, it ruptures to release the secondary oocyte, that is the ovum, from the ovary, and this process, it is known as the ovulation. So now let's move on to the next slide. So it is very important to understand the difference between the spermatogenesis and the oogenesis. Now let's go through the points. So the spermatogenesis, it is occurring in the testis, and the oogenesis, it is occurring in the ovary. So there is a limited growth phase in the case of the spermatogenesis, whereas there is an elaborated growth phase in the case of oogenesis. So each primary spermatozoite, it is responsible for giving rise to 4 sperms, whereas each primary oocyte, it gives rise to a single ovum. So this difference, it is very important. So there is no polar body formation in the case of the spermatogenesis, whereas polar bodies, they are formed in the case of the oogenesis. So the spermatogenesis, it is beginning at the puberty and it extends up to the old age, whereas it begins in the case of the embryonic stage but is suspends at the puberty. So it gets arrested at the prophase 1 of the meiosis. Then it, when after the puberty, when the menstruation it is starting, till that is the first menstruation, it is known as the menarche, and it ceases around the age of 50, that is known as the menopause. So no development of the the gamete it is happening after the menopause, that is the last menstrual cycle that is happening till the age of 50. So now let's move on to the next slide. So menstrual cycle, this is also a very important concept, and surely questions will be asked from this topic. So let's go through it. So the cyclic events in the female reproductive system from one menstruation till the next during the reproductive puberty, that is the puberty to menopause, it is known as the menstrual cycle. So the duration of the menstrual cycle, it is 28 or 29 days, and it has the following 4 phases that is happening. So it first phase, it is known as the menstrual phase, that happens during the 1 to 5th, it happens in between the 1 to 5th day of the menstrual cycle. And the main events that are happening in this phase is that the menstrual flow it happens, that is known as the bleeding. So follicular, that is also known as the proliferative phase, it happens during the 5th to 13th day of the menstrual cycle. So in this, the primary follicles, they get developed into the Graafian follicles, and proliferation, it means that the rapid cell division it is happening of the ruptured uterine endometrium. So in the third stage, it is the ovulatory phase, in which it has happening in the 14th day. So the luteinizing hormone, it undergoes a sudden surge before the ovulation. So it happens prior to the ovulation. So there is a surge in the luteinizing hormone, the rupture of the Graafian follicle it happens, and the ovum it is getting released from the Graafian follicle. The process it is known as the ovulation. So LH it is surging before the ovulation process. This point it is very important. So the last the phase, it is the luteal phase or the secretory phase, that happens during the 15 to 28th day of the menstrual cycle, in which the corpus luteum it is forming, and the formation of the corpus luteum it leads to the secretion of the progesterone. So this progesterone hormone it helps in the uterine thickening, and it helps in the to make it vascular and also soft. So the first menstruation that is happening during puberty, it is known as the menarche. Whereas the last menstrual cycle, that is the permanent stopping of the menstrual cycle, at the age of the 50, it is known as the menopause. So this is very important, and we need to know this. So now let's move on to the next slide. So the hormones that are involved in the menstrual cycle, we need to know the role and the gland that is getting involved in the secretion of those hormones. So this is very important, and questions can definitely be asked from this. So the endocrine gland, we have the anterior pituitary and the ovaries that are producing the hormones of the menstrual cycle. So the hormones that are produced by the pituitary are anterior pituitary, it is the follicle stimulating hormone and the luteinizing hormone, and the hormones that are produced by the ovaries, it is the estrogen and the progesterone. So now let's us understand the role of follicular stimulating hormone and the luteinizing hormone. So follicular stimulating hormone, it is involved with the stimulating the follicular growth in the ovaries. So from the primary follicle till the Graafian follicle, it is the growth of the follicle, it is getting stimulated with the help of the follicle stimulating hormone. The name in itself, it is suggesting us that it is helping in the follicular growth in the ovaries. So it helps in the stimulation to secrete the estrogen. So the role of the luteinizing hormone, let us discuss that. So the surge of the luteinizing hormone that happens prior to the ovulation, it helps in the ovulation process. So the LH surge, it causes the ovulation, that is the graphene follicles it ruptures and the ovum it is getting ruptured out. So the it results in the formation of the corpus luteum. So luteinizing hormone it results in the formation of the corpus luteum. So the corpus luteum it is responsible for the secretion of the progesterone. So now let's go through the hormones that are secreted by the ovaries. So first one it is the estrogen. So estrogen it is responsible for thickening the uterine lining, that is the endometrium, and it inhibits the FHS and the LH for most of the cycles. So the same two role it is of the progesterone also, that it helps in the thickening of the uterine lining, that is also known as endometrium, and it inhibits the FSH and the LH for most of the cycle. So the estrogen it is also involved in the stimulating the FSH and LH before the pre-ovulation. So before the ovulation, it stimulates the formation of the FSH and LH, that is done by the estrogen. So now we have studied the role of the hormones that are taking part in the menstrual cycle. So now let us move to the next key concept from this chapter. So this graph, this is a very important topic, and sure shot a question will be there from this graph itself in the exam. So now let us try to understand this graph. So here we have the pituitary hormones, that is the FH and the LH, and here we have the ovarian hormone, that is the estrogen and the progesterone. So we know that in the developing follicle, we know that estrogen it is helping in the development of this follicles. So what is happening is that, now let us go through the first point, the secretion of the FH and the LH. So here we can see that the FH and LH they are both getting secreted during the follicular phase. So they stimulate the development of the follicles. So we can see here that these secretions it is helping in the development of these follicles. From the primary follicle it is getting developed till the mature follicle. So the FH and LH it is responsible for the development of these follicles. So estrogen it is stimulating the proliferation of the endometrium. So here we can see that the estrogen as it is getting increased, it stimulates the proliferation of the endometrium. So the level of LH and FH it is peaking in the middle of the cycle. So here we can see that this is the middle of the cycle, and here both the FH and LSH it is getting peaked. So the LH it undergoes a surge. So here we can see that the LH it has undergone a sudden surge before the ovulation process. So here we can see that the mature follicle it has ruptured and the ovum it is released out from the follicle. So the LH it is getting surged before this ovulation. So after the ovulation, the ruptured follicle, so here the follicle it has been ruptured. So what happens with it? So it gets developed into the corpus luteum. So this corpus luteum it is then secreting here the progesterone. So the developing corpus luteum it is responsible for the secretion of the progesterone here. So the progesterone it is responsible for the maintenance of endometrium for the implantation process. So in the absence of fertilization, so if fertilization is not happening, this corpus luteum it starts regressing. So what happens is that it regresses and the progesterone level it is also getting decreased as it is getting regressed. So it leads to the disintegration of the endometrium. So here we have the uterine events. So this graph here it is showing the uterine events, the uterine lining events, that is the endometrium. So it leads to the disintegration of the endometrium, and thus the menstrual flow then again it is occurring. So now let's move on to the next concept. So fertilization and implantation. Now let's go through it. So in this slide we are going through the process of the fertilization. So here the sperms, they are traveling inside the vagina through the cervical canal, the uterus, the isthmus. They finally reach the ampullary region of the fallopian tube. Also, the ovum from the ovary, it travels to the fimbriae, then to the infundibulum, and then then it reaches the ampullary region. So both the sperm and the ovum, when they reach the ampullary region at the same time, so the fusion of the sperm and ovum, it is happening, and it is happening in the ampullary region of the fallopian tube. So this fusion, it leads to the formation of the zygote. So this whole process, it is the fertilization process that is in detail. So now let's move on to the next concept. So the next concept is of the implantation process. So the zygote, it is formed after the fusion of the male and female gamete. Now what happens with the zygote? Let us understand. So here we can see that the zygote, it has formed. So after the formation of the zygote, it undergoes cleavage. So it starts getting dividing here. We can see that it is getting divided into this mass of cells. So after a quite few divisions, it leads to the formation of the morula. So this is very important point, that the morula, it consists of the 8 to 16 blastomeres. So blastomeres, they are a group of the dividing cells. So here we can see that the morula, it is formed, and it consists of the 8 to 16 blastomeres. Okay, and after that, this morula, it leads to the formation of the blastocyst, which then forms the embryo. So here we can see that this is the blastocyst diagram here. So here the inner mass of the cells, it leads to the formation of the embryo, and this outer mass of cells, they provide nutrition to the embryo. So the attachment of the the blastocyst is then embedded into the uterine wall, that is known as the endometrium. So this embedding of the blastocyst into the uterine wall, it is known as the implantation process. So now let's move on to the next slide. So placenta, what is the placenta? Let us understand. So placenta, it is the structural and the functional unit between the fetus and the uterine wall. So it is formed by the interlocking of the craniovilli and the uterine tissue. So the placenta, it is the structural and functional unit between the fetus and the uterine wall. So the functions of the placenta, it is very important, and questions will be definitely, it can come from this. So it is responsible for supplying oxygen and nutrients from the mother to the fetus. So it also helps in removing the carbon dioxide and excretory waste from the fetus. So it is acting as a endocrine gland. So it is responsible for producing the human chorionic gonadotrophin, the human placental lactogen, the estrogen, the progesterone, and relaxin. So this point, it is also very important, that the hormones that are produced from the placenta, it is very important. So 4 hormones can be given, in which one hormone, it can be LH or FSH, that is developed from the pituitary gland, and 3 hormones can be given from the placenta, and a question can be asked, which is the pituitary hormone? So we need to take the FSH or the LH. Whereas all these hormones, they are the hormones of the endocrine gland, that are the placental hormones. So now let us move to the next key concept from this chapter. So parturition and lactation. So these processes, they are also very important. Now let's go through it. So some signals are given from the fetus and placenta, and it leads to the mild uterine contractions. So these contractions of the uterine, they are known as the fetal ejection reflex, and this it causes the release of oxytocin from the pituitary gland. So when the oxytocin, it is getting released from the pituitary, it then leads to stronger uterine muscle contractions, which further leads to secretion of the oxytocin, and thus the parturition process, it happens, that is giving birth to the baby. So lactation, it is the production of the milk from the mammary glands, and colostrum. So in the initial days, the mother's milk, it would be very yellowish color, in very yellowish color, and it would be and it would consist of antibodies that are required to keep the baby safe from the pathogens that are present in the environment, because the immune system of the baby, it is developing in the initial stages, in initial days after the birth. So the antibodies, it is the baby, it is getting antibodies from the mother's milk, that is known as the colostrum, and it is yellow in color. So now let us move to the next slide. So we have covered all the important concepts from the chapter 3. Now let us go through the main concept from the chapter 4, that is the reproductive health. So in this chapter, a very important process that is known as the amniocentesis. So what is happening in this process? Let us understand it. So in this process, the amniotic fluid of the fetus, it is taken out and it is analyzed. And it was originally done in order to know whether the baby, it is having some kind of genetic defect, or it is having any type of diseases. So that is why it was done. So it helps to check the chromosomes of the baby, and thus after the checking we get to know whether it is having any kind of infections or genetic defect. So that is why this process was done. But what happened is that this process, it also helped in revealing the sex of the fetus. So as it revealed the sex of the fetus, the parents, they could know whether they would have a female or a male child. So thus it had to be banned in India, because the female child, it is not accepted in the many parts of our country. So if the sex of the fetus, the parents, they know that they would have a female child, then the female fetus was aborted, and thus this amniocentesis process, it needed to be banned in India, because it could reveal the sex of the fetus. So a Saheli, it is the oral contraceptive which is used by the females. It was developed by the Central Drug Research Institute that is present in Lucknow in India. So this so by contraceptive means that it helps in the prevention of pregnancy. So it is a oral pill that helped in the prevention of pregnancy. So now let us move to the next key concept from this chapter. So now let us understand various other methods of contraception. So first we are going through the natural and the traditional methods of the contraception, in which the main aim is to avoid the chance of the ovum and the sperms meeting. We are avoiding any chances of the meeting of the ovum and the sperms. So in this process, in this method, we have three processes. So let us go through it. So first one, it is the periodic abstinence process. So in this process, the couples, they avoid sex from day 10 to 17 of the menstrual cycle, when ovulation, it can be expected. So that is known as the periodic abstinence. So the second method, it is the withdrawal or the coitus interruptus, in which the male partner, it withdraws the penis from the vagina just before the ejaculation. So this helps in avoiding the insemination. So lactational amenorrhea is also a type of natural or traditional method to prevent pregnancy, in which what happens is that there is the ovulation and therefore the menstrual cycle, it doesn't happen during the period of the intense lactation following the childbirth. So the intense lactation after the childbirth, it lasts for around 6 months. So at that time, the ovulation and the menstrual cycle, they do not happen. So as long as the mother is breastfeeding the child fully, the chances of the conception, it is almost nil. So it is effective for the 6 months. So now let's go through the next topic. So barriers. So in barriers, we are using they are barriers. They are the physical methods in which we are preventing the ovum and the sperms from meeting. So they are physical methods, right? So in which the first one, it is the condom, which is a rubber or a latex sheath that is worn over the penis in the case of males, and it is worn in the cervix region in the case of females, that helps in the prevention of the from semen in entering the, so the ejaculate semen, it cannot enter into the female reproductive tract. So Nirod, it is a popular brand of the condom. So the use of condoms, it also helps in protecting from various sexually transmitted infections and also from AIDS, and it also helps from getting pregnant, right? So the other mode of barriers, it is diaphragm, cervical caps and vaults. So barriers, they are made up of rubber that are inserted into the female reproductive tract to cover the cervix during the coitus. So they prevent the conception by blocking the entry of sperms through the cervix in the similar manner of condoms, right? So spermicidal creams, jellies and foams, they are also used along with these barriers to increase the contraceptive efficiency. So intrauterine device. So this is very important. A question can come from this. So there are non-medical IUDs such as lip loop, copper releasing IUDs such as CU7CUT multilode 375, and the hormone releasing IUD, that is progesterone and LNG20. So this, from this, a match the following question can be come. So IUDs, intrauterine device, they increase the phagocytosis of the sperms. So within the uterus, and the copper ions, they suppress the sperm motility and the fertilizing capacity of the sperms. So the hormone releasing IUDs, they make the uterus unsuitable for the implantation process, and the cervix, it remains hostile to the sperms. So now let us move to the next concept. So oral contraceptives. Let's go through this topic. So oral administration of the small doses of either progesterones or the progestogen estrogen combinations. So the hormone name we need to know, that is progestrogens or the progestogen and estrogen combination, is another contraceptive method that is used by the females. So they are used in the form of tablets, and hence they are also known as the pills. So pills, they inhibit ovulation and implantation, as well as they alter the quality of the cervical mucus to prevent and retard the entry of the sperms. So pills, they are able to inhibit the ovulation, they are able to inhibit the implantation, and also they alter the quality of the cervical mucus that helps in prevention of the entry of the sperms. So these pills, they are also known as the emergency contraceptives, as they could be used to avoid the possible pregnancy due to rape or casual unprotected intercourse. So now let us move to the next concept. So surgical methods are they are generally irreversible methods. So when the couple, they decide that they do not want to have any children any further, so you go for these type of methods that are known as the surgical methods. So these methods are generally irreversible. Once they happen, they cannot be reversed back. So in the males, what is happening, this method, it is known as the surgical method, it is known as vasectomy, and in the case of females, it is known as the tubectomy. So in vasectomy, what is happening is that the vas deferens, they are tied and they are cut. So the movement of the sperms, it doesn't happen because the vas deferens, they are cut from here in between. So in the case of tubectomy, the fallopian tubes, they are cut. So these methods, they are generally irreversible. So now let's move on to the next slide. So don't get confused. So vasectomy, it is done in the case of males, and the tubectomy, it is done in the case of females. So don't get confused that you choose, in the case of males, you choose tubectomy. So that must not happen. Just try to remember is that vasectomy, the starting letter, that is V, it indicates vas deferens. And vas deferens, we know that it is present in the males, right? That is cut, tied and cut in the case of the vasectomy. So try to remember like this so that you don't get confused. So now let's move on to the next slide. So medical termination of pregnancy, it is abbreviated as MTP. So the intentional or the voluntary termination of pregnancy before full term, it is known as the medical termination of pregnancy, or it is also known as induced abortion. So why the medical termination of pregnancy? It is needed. So it is needed in order to get rid of the unwanted pregnancies. So if the couple is not planning a baby, so they can get rid of the unwanted pregnancy by using this medical termination of pregnancy. So so if the pregnancy it leads to harm to the health of the mother or fetus or both, then this process it can be done so that no harm is done to the baby or the mother. So MTPs, they are considered safe during the first trimester, that is up to 12 weeks of pregnancy. After that it can cause harm. So it is MTPs, it is considered safe in the first trimester, that is up to 12 weeks of the pregnancy. So the misuse of medical termination of pregnancy, it is being done. So medical termination of pregnancy, it is getting performed illegally by unqualified persons, which is unsafe and dangerous. So this process, it needs to be performed strictly by the medical professionals and doctors only. Otherwise it can cause harm to the mother, so or the baby also. So the fetus, if it is found to be female, like we had learned in the case of amniocentesis, then by the process, then it is then aborted by this medical termination of pregnancy process. So now let us move to the next concept. So infertility. So what is meant by infertility? It is the inability to conceive or produce children even after having 2 years of unprotected sex. Then it is known as infertility. So the infertility, we can use several technologies in order to correct this infertility, and we can have baby. So this is done by the use of the assisted reproductive technology, that is abbreviated as ART. So in this process, in the ART technologies, the various processes they are present. So one by one we would be going through all of these processes, as match the following question can be come from this topic. So we have the gamete intrafallopian transfer, the intracytoplasmic sperm injection, the artificial insemination, in surrogacy, in vitro fertilization, that is also known as the test tube baby program. So now let us start with all these ART technologies, right? So first one, it is the in vitro fertilization, that is also known as the test tube baby program. So what is the methods in that is taking place in this process? Let us go through it. So the egg, it is collected from the female donor, and the sperm, it is collected from the male donor, and they are both made to fuse in the laboratory conditions. So in vitro, it means inside the lab it is happening, in vitro. So the fertilization, when it happens, the zygote is formed, then it leads to the formation of embryos, and then it is transferred into the uterus or the fallopian tube of the female, right? So what is happening is that the embryo transfer, it can happen in the two ways. So first way, it is the zygote intrafallopian transfer. Another one, it is the intrauterine transfer. Now let us go through both these embryo transfer processes, right? So what is happening in the case of zygote intrafallopian transfer is that the transfer of zygote or the early embryos up to the 8 blastomeres, that is the keyword here, it is taking place in the fallopian tubes. So in zygote intrafallopian transfer, when it is up to 8 blastomeres, we are transferring it into the fallopian tube. Whereas in the case of intrauterine transfer, the transfer of embryos with more than 8 blastomeres, it is transferred into the uterus. So intrauterine transfer, more than 8 blastomeres is getting transferred. Whereas in the case of ZIFT, up to 8 blastomeres, it is getting transferred, right? So embryos that are formed by in vitro fertilization, that is the fusion of gametes within the female, it also could be used for such transfer to assist those females who cannot conceive. So now let us move to the next assisted reproductive technology process. So gamete intrafallopian transfer, it is done when the female cannot produce ovum, but it can provide a suitable environment for a ovum that is received from a healthy donor. So the affected individual cannot produce a ovum, but it can provide a condition for the growth of a ovum that is received from the donor. So that is this process, it is done at that time. So now let us move to the next technique. So in intracytoplasmic sperm injection, what is happening is that the sperm, it is directly fused with the ovum, that is the egg cell, in the laboratory type of conditions, and the embryo then it is implanted into the woman's uterus. So now let us move to the next important concept. So the artificial insemination technique, it happens when the number of sperms that are produced by the male in the semen, it is quite low, or whether the male partner is not able to inseminate the female. At that case, this technique, that is the artificial insemination technique, it is done. So what is done is that the sperm, it is taken from the healthy male donor and it is injected into the uterus of the female. So now let us move to the next concept. So we have discussed all the key concepts from the Unit 1 of the CUET entrance syllabus for biology. I hope that all of these concepts help you in your speedy revision, and we would be revising in such a manner all the other remaining units from the CUET entrance syllabus. So thank you for your patient listening and your valuable time.